Hello, my name is Lynn. This assignment video is for microbiology class, and the topic is about choosing a microorganism that we think is interesting or special, or that it possesses a special characteristic. And the microorganism that I chose for this project is a radioactive resistant bacteria, Dinococcus radiodurans. And I hope those pictures explain the name itself. So I think as you go further into the slides, I think it makes you realize that scientific discovery do occur unexpectedly, and I think the discovery of Dinococcus radiodurans falls within this exception, or that it is a great example of this statement. Okay, so Dinococcus radiodurans were discovered by a scientist named Arthur W. Anderson. So what he tried to do was he tried to sterilize this can of meat through ionization radiation. And eventually, what happens at the end was that the can of meat, the meat inside the can, spoil. So what that means was that what happened was, uh, I'm sorry. So this proves that there are bacteria in this can of meat, and that's how Dinococcus radiorans were discovered. And what's remarkable about this bacteria is that it has the ability to repair DNA damage. It's also resistant to desiccation and also UV radiation. And ionization radiation. So it takes about 5 to 10 grays of radiation to kill a human being. Grays is a unit for radiation. And also E. coli, it takes about 200 to 800 grays of radiation to kill E. coli. But for Dinococcus radiodurans, it takes about 5,000 grays of radiation to kill this bacteria. So this is one tough bacteria. It takes thousands of times more to kill this bacteria than to kill other bacteria or even a human being. So if there's a nuclear war going on, this is definitely the only time you would want to be a bacteria, especially this one. Okay, so ecology and morphology. So Dinococcus radiodurans is a coccus, meaning that its shape is oval and spherical. It is a gram-positive bacteria, meaning that it has a thick peptidoglycan. Always have a hard time saying that word, peptidoglycan. Okay, I gotta say it real fast, but anyways, so it's a gram-positive bacteria. It doesn't have an outer membrane. Okay, it has a thick peptidoglycan. Um, its structure is a tetrad formation, meaning that it has two planes, each plane containing cell nucleus, and the nucleus has DNA on both sides of the planes. And this bacteria is non-sporulating, meaning that it doesn't produce endospore when the environment is inhospitable or lethal, um, or that it is a stressful environment and it doesn't produce endospore. So the bacteria is an obligate aerobes. So what this means is that it needs oxygen to grow. So they require oxygen for growth. So what happened is that they use oxygen for producing energy and respiration. Okay, so it all this process depends on the transfer of electrons to oxygen, which is the final electron acceptor in the electron transport reaction. Okay, so you can find these bacteria in feces, soil, sewage, meat. It is a polyextremophile, so it can survive in any weather: cold, hot, dehydration, and even acid. So these are the metabolism pathway for Dinococcus radiodurans. It uses pentophosphate pathway and glycolysis. So Deinococcus radiorans is a chemo-organoheterotrophs. Okay, so it requires an organic substrate to get its carbon for growth and development, and it also produces its energy from oxidation and reduction of an organic compound, such as glucose. Okay, so it uses several pathways, such as glycolysis, pentophosphate pathway, for the breakdown of glucose. Okay, so these pathways are also vital for the recovery from DNA damage agents such as oxidative stress, free radical, which produced by ROS, reactive oxygen species. The pentose phosphate pathway, what happened is they convert glucose into DNTPs. Okay, so DNTP stands for the deoxynucleotide triphosphate. So these are nucleotide bases that can be used for new DNA strands during replication. So glucose, they provide substrates for DNA repair and protection against ROS toxicity. So once again, Deinococcus radiorans can survive radiation, extreme environmental conditions through its ability to repair DNA and eliminate ROS from the cells. So what happened is DNA damage, right, through radiation causes these double-stranded breaks called DSBs. And DSBs are much harder to repair than SSB. SSB can easily be repaired by DNA polymerase 1 and DNA ligase as opposed to DSBs. Double-stranded breaks, or DSB, in order to repair them, the steps are more complicated. The mechanism is more complicated. The proteins and enzyme, there's just much more involved. Okay, so and also the ROS, these reactive oxygen species, are caused by UV radiation, desiccation, and ionization radiation. So what happened is that these ROS species, they damage the proteins, they damage the DNAs, and they damage the lipids. But good thing is the bacteria are able to repair DNA fast and efficient. Okay, so this is just a nutshell of the mechanism and the key players in the repair mechanism of Deinococcus DNA repair. I would just try to explain this in a nutshell. It's a little complicated, but for this diagram is just a shorter version of the longer mechanism. Okay, so let's start out. The process begins with ESDSA, which stands for extended synthesis dependent strand annealing. Okay, and then followed by DNA recombination. So the ESDSA process is done through the first phase with the nuclease and the DNA polymerase function. So this is where the excision repair is done to the DNA. Homologous recombination means the broken chromosome is reattached to the homologous chromosome or to the partially replicated chromosome. The REC-BCD enzyme helicase or nuclease binds to the double-stranded DNA and initially degrades the DNA using its exonuclease. So for phase 2, the REC-BCD enzyme then loads the REC-A proteins onto the 3' terminated strand, and REC-A can carry out strand invasion of the homologous duplex to initiate the recombination reaction. Okay, so last but not least, the importance of Deinococcus radiodurans. Why do we need to study them? Well, they have an amazing ability to repair DNA in a fast and efficient way. Um, they have the ability to eliminate ROS, reactive oxygen species. And if we can learn more about their mechanism, then we can definitely apply it to biotechnology to help cure diseases, cancers, um, and just overall make our life better. So this is the end of the project.